So when we look at bacteria, single-celled, no organelles, no nuclei, but then why do we even care about them if they're kind of at the bottom rung of evolution, so to speak, as far as living organisms? Well, we've seen how they're classified. We've got two major domains that are these bacteria type or microbe type, okay, simple organisms. They produce about a half of the Earth's oxygen. Regardless of plants, they would still be producing that very, very high level of oxygen for us. They do all nitrogen fixation. We've looked at this before, the bacteria in the nodules of legumes and so on. Critical for decomposition. We wouldn't be able to walk around this planet. It would be covered in trash, garbage, dead organisms if it weren't for the bacteria and microbes that were breaking them down. And on the Earth itself, we look at half of the Earth's biomass. So remember, biomass is carbon. So half of the carbon on this entire planet, including the blue whales and the trees and you, half of the Earth's biomass is microbes. And if we look at just you have more bacterial cells than human cells in your body, and you never will have more human cells than bacteria in your body. While we look at this as kind of being yucky, we also have to think about them having a very significant role for you. So when we look at our bacteria, they've got a couple of characteristics that we need to consider. Okay, so we said there's no nucleus. Okay, there are no organelles. Okay, there's no cytoskeleton. So remember, cytoskeleton was structural for animal cells and plant cells and so on. Okay, we do see DNA present, but it's a single circular chromosome. And they reproduce by something called binary fission. So these are big characteristics for our bacteria or microbes. Now, when we look at them in relative size structure, the bacteria are fairly small, about 2 micrometers. Viruses that we talked about before are about 1/10 of that size. And when we get into protists and actually looking at animal kingdom sorts of things, then we start looking at these protists, which are ancestors to the animal kingdom. They are much, much larger, as you see. So we've got to think about relative scale, particularly when we talked about a minute ago the idea that they compose such huge quantities of biomass on this planet. Now, bacteria come in a couple of shapes. They are named based on their shape. So we have cocci, which are spherical, bacilli, rod-shaped, and spirochetes, kind of these spirally S-shaped ones down here. Now, you may have heard of bacillus before. So a bacillus would be a rod shape, okay? Some of you may have had staphylococcus, strep throat. Coccus means that it's spherical, so it's a bacteria that infects that's spherical in shape. When we actually look at how they infect, they're going to have a coating around the outside or a capsule around the outside that allows them to stick to target cells. Now, for these capsules to stick to and adhere to these target cells, it allows them to very quickly infect organisms. But when we look at their infection, most of the time it's not the bacteria itself that's dangerous, it's the toxin or the waste that the bacteria produces that actually makes you sick, that causes the infection. So prokaryotes, because they are single-celled, they're replicating roughly every 20 minutes. They can have extremely high population growth rates. So this means in many cases we have very high evolution rates. Okay, we've talked before that we actually don't even bother naming species. Remember, we talk about strains of bacteria because they just replicate so fast we can't name all of the species associated with them. So we said by binary fission they're going to divide and produce new generations within hours. Typically we're looking at about 20 minutes per generation. And huge amounts of that genetic variability, as we mentioned. Now, they are going to suffer from mutations. They don't have DNA repair mechanisms. So this is actually going to lead to even higher levels of evolution for us. And we look at them, because of these high mutation rates, at being excellent survivors. So with changes in the environment, they can have organisms, individuals that will survive even the biggest changes in the environment. So we've talked before that we can actually look at extremophiles or the archaea as being important, an important domain for us. Still prokaryotes, still bacteria in a sense, but we can look at all bacteria as really being high-level survivors in environments. The genome itself, about 1/1000th the DNA that a eukaryotic genome would have. It is circular, as we noted, and it is not contained in a nucleus, but rather is contained in an area known as the nucleoid region. We can also look later on at a number of modifications to prokaryotes. They tend to be kind of greedy about their environment, and they pick up DNA readily from their environment. And when they do that, they can actually incorporate as something known as a plasmid into their chromosome. This will become very important when we talk about genetic engineering. So these plasmids get incorporated potentially into the chromosome of the bacteria and increase variability and actually make that bacteria even less susceptible to change in the environment and can have very beneficial effects as far as survival and reproduction. Now, in the environment itself, we've talked about nitrogen-fixing bacteria before, associated with legumes. We look at a number of them being critical for your diet, particularly in your small intestine, and helping you to digest foods at a higher level than you normally would. And then also as decomposers. So we mentioned the idea that their role in decomposing and breaking down waste on our planet is critical. As far as you, in your mouth alone, you have about 600 species of bacteria, and the number of bacteria in your mouth is greater right now, today, than the number of people that have ever lived on Earth. Okay? So these are huge numbers of bacteria, but it's okay. Don't worry. In your digestive tract, you have about 100 trillion bacteria, and if you actually were interested in drying out and weighing your feces, the dry weight of your feces is actually about 25% bacteria by weight. So these are huge numbers of bacteria. But they play a very critical role in your body. Now, they're not all good, right? But when we look at the ones associated with your digestive tract, they actually did some studies looking at rats and the bacteria in their digestive tract. They eliminated all of the bacteria in their digestive tract with antibiotics and studied the diet of the rats, and it turned out they needed 30% more calories to maintain their body weight without those bacteria. So they require, just as you do, those bacteria for healthy digestion and incorporation of materials into the body and absorption of materials into the body. There are lots of sugars and vitamins that we would not be able to use or make without bacteria. We see some bacteria here, cocci in particular, because they're round on the surface of the cells of your intestine. So these are nice long intestinal cells down here. We call them columnar cells. Columnar epithelial cells line your intestine, and the bacteria sit on the tops of them, helping you to absorb. Now, they're not all nice, right? Some bacteria make you very, very sick. As we mentioned, most of the damage comes with the toxins or the waste products that are secreted by the bacteria. So when we look at those kinds of bacteria, we actually are looking to fight them typically with antibiotics. So the antibiotics that we use were developed a very, very long time ago, and our concern now is that many of our bacteria that we're exposed to are actually what we call antibiotic resistant, meaning that the bacteria are no longer eliminated or affected by the antibiotics. So these antibiotics that are produced, penicillin, things like that, may not be strong enough to kill the bacteria. The bacteria are now very strong, and we'll look at some strains of these antibacterial or antibiotic resistant bacteria in the future. So when we look at actually the discovery of penicillin, it occurred about 200 years ago now, looking at its origins actually from fungus. It was actually a lab mistake. For those of you who are interested, it's actually a fun story. And penicillin actually is a derivative of bread mold. And bread mold is a fungus. So we actually look at penicillin discovery based on an extract from bread molds.